Good day everyone, I am Jobert Rondina. I am the next reporter in chapter 9. So my topic is all about genetic modification. So genetic modification is also called generic engineering and the uses of GMO. So what is genetic engineering? So genetic engineering refers to the science of identifying, isolating, and alternating of genes. The purpose of altering the genetic makeup of an organism is to come up with one or more desired traits that are not found originally in a certain organisms. In this process, the genes are removed using a technique called gene splicing. Genetic engineering is also called genetic modification. The product of genetic engineering is called genetically modified organisms or GMOs. So, through these lessons, we learn to understand how genetic works, to define an altering of genes. So, we'll know DNA, or also called deoxyribonucleic acid, or DNA, refers to the molecule found in nucleus of every cell. This molecule made up of four subunits which are represented by letter A, C, G, and T. So, ang A stands for adenine, C for cytosine, G for guanine, and T for thymine. These subgroups are nucleotides which are responsible for productions of proteins that are needed for the organisms to grow and live. The segments of DNA are called genes. These hold instructions on how to produce a single protein. Each organism consists of thousands of genes. The set of genes in an organism is called genome. DNA is known as universal language. This means that the genetic code of all organisms is the same when a gene for consisting of a desirable trait is taken from one organism and it's inserted into another. The recipient of gene will acquire an ability to express the trait in code. While proteins, these are the biomicromolecules which are responsible in all works done by cell. They are also part of the structures such as a cell wall, organelles, and tissues. The macromolecules regulate the reactions that take place in the cell. They also serve as enzymes which speed up reactions. Also, these lessons, we learn the process of genetic engineering. The alterations of genes was also done using biotechnology. So, what is biotechnology? So, biotechnology refers to the use of biological techniques and engineered organisms to make products of our plants and animals that have desired traits. To carry on this technology, it employs different processes, such as first is the restriction enzymes, second, gel electrophoresis, third, recombinant or DNA technology, fourth, gene cloning, fifth, DNA sequencing, the last, polymerase chain reactions are also called PCR. Analytical genetic engineering, a research branch of genetic engineering in which virtual genetic models are created using computer software. This type of engineering is a better way of carrying out the trial and error of the study since it reduces risk of disaster during experiments using real organisms. First, chemical genetic engineering. This type of engineering deals with the separating, classifying, and grouping genes and preparations for applied genetic engineering activities and experiments. Second, genetic mapping refers to the process in which DNA fragments are assigned to individual chromosomes. The genetic map is created after completing DNA sequencing. Genetic mapping is very important in understanding a disease gene length. Genetic interactions, on the other hand, helps the researchers understand exactly what set and combination of genes would produce a particular phenotype or set or morphological, psychological, and behavioral characteristics. Gene coding deals with the study and conduct experiments with amino acids, sequences of DNA, and RNA. Applied genetic engineering This refers to the field of genetic engineering, which pertains to the practical applications of genetic engineering tools manipulate the genes of living organisms to make genetic couples. This includes two setups. First, one is what we call as cloning, and the second one is the transgenesis. There are three types of genetic modification. First, plant genetic modification. It is a selection is considered as the easiest method of plant genetic modification. Genetically, heterogeneous populations of plants is inspected, and plants with the most desired traits like improved palatability and yield are selected for continued propagation. This is used to produce pest-resistant plants and increase crop of production. Second, animal genetic modification. The established and emerging biotechnology in animal agriculture greatly helps the productions of quality livestock. This includes assisted reproductive technologies use of naturally occurring hormones such as recombinants and biotechnologies to enhance reproductive efficiency without affecting the genome, biotechnologies to enhance expression of desirable genes, for examples, increase of milk productions per cow, second double productions of eggs and chickens, and third, genetic modifications of microorganisms have significant roles in food production. They have primary and secondary role in food fermentations and food spoilage. They can also produce enzymes or other metabolites used in food productions and processing. Humans have used genetically modified microbes for centuries to produce food, wine, bread, and cheese. The following are examples of how genetic modifications of microbes greatly help the people. For examples, increase microbe-dependent food production. Second, increase human immunity resistance to microbe-caused diseases. These are the advantages, benefits of genetic engineering. The following are the possible benefits of genetic engineering. First, more nutritious. Second, tastier food. Third, less use of pesticides. Fourth, faster growing plants and animals. And also the advantages of human genetic engineering. Genetic engineering could further human lifespans. Second, we could rid of hereditary disease. Third, every child would have chance to be born healthy. And also there are the disadvantages of human genetic engineering. First, there will always be ethical questions to consider. Second, it would change how we would need to approach a population's control. This is my second topic in chapter 9. It's all about the Cartagena Protocol on Biosafety. So, the Cartagena Protocol on Biosafety to the Convention on Biological Diversity, CBD, is an international treaty government of living modified organisms or LMOS, resulting from modern biotechnology have from one country to another. The protocol has been developed in response to advancement in the area of modern biotechnology may have negative effects on biodiversity and human health. It is an attempt to produce globally harmonized regime for biosafety to ensure safe use of modern biotechnology. The Convention on Biotechnology The Convention on Biological Diversity was finalized in Nairobi in May 1992, opened for signature at the United Nations Conference on Environment and Development or UNCED in Rio de Janeiro on 5th July 1992. It entered into force on 29 December 1993. Today, the convention is the main international instrument for addressing biodiversity issues. It provides a comprehensive and holistic approach to the conservation of biological diversity. Biosafety is one of the issues addressed by the convention. This concept refers to the need to protect human health and environment from possible adverse effects for the product of modern biotechnology. At the same time, modern biotechnology is recognized as having a great potential for the promotions of human well-being, particularly in meeting critical needs for food and agriculture and healthcare. This is the last topic that I will discuss in chapter 9. It's all about genetically modified golden rice falls short on life-saving promises. GMO activists not to be blamed for scientific challenges slowing introduction. Study finds heralded on the cover of Time magazine in 2000 has a genetically modified crop with the potential to save millions of lives in the third world. Golden rice is still years away from field introductions and even then may fall short of lofty health benefits still cited regularly by GMO advocates suggest a new study from Washington University in St. Louis. Golden rice is still not ready for the market, but we find the support for the common claim that environmental activists are responsible for installing its introduction. GMO opponents have not been the problem, said Lindenwood author Glenn Stone, professor of anthropology and environmental studies in arts and sciences at the University of first conceived in the 1980s and a focus of research since 1992. Golden rice has been a lightning rod in the battle over genetically modified crops. GMO advocates have long touted the innovation has a practical way to provide poor farmers in remote areas with a subsistence crop capable of adding much needed vitamin A to local diets. A problem in many poor countries in the global south, vitamin A deficiencies leave millions on high risk for infection, diseases, and other maladies such as blindness.